adipose-derived stem cell secretome group, in particular, demonstrated remarkable effectiveness in enhancing hair density and promoting the initiation of the anagen phase, the active growth phase of hair follicles. Notably, this group achieved these results without inducing the common side effects associated with topical minoxidil use, such as the typical scalp irritation and dryness, which probably comes from the alcohol and the propylene glycol. But in any case, this highlights to us that the adipose-derived stem cells are a potent standalone option in the realm of regenerative therapy for hair loss, and it shows a great promise in both efficacy and safety. I know there are some clinics out there that are like, hey, we have special exosomes that, that can grow your hair. I mean, always be skeptical about these things and actually look at what those exosomes are, how they're harvesting them, and don't become a sucker. Hello everyone, and welcome back to yet another video. Today we're going to be looking at a pretty cool study involving secretomes and their possible implications for stimulating hair growth. It is in my personal opinion that secretome research will lead us to discovering novel ways in repairing tissues as well as restoring organ function. And because the hair follicle is an organ, and in various alopecia conditions, this organ is either suppressed to the point where it enters a dormant-like state, like we see in androgenetic alopecia's end stage, or the hair follicle is destroyed like we see in autoimmune, cicatricial, or scarring alopecias like lichen planopilaris. In both of these cases, secretomes may allow us to restore these mini organs, and even in some cases, regenerate them. But before we look at any study, what is a secretome? Secretomes are proteins and other factors secreted by a cell, hence the Latin root word secret, meaning to be moved apart or separate. This term was first created by Schalzma et al. In 2000 in a paper titled, quote, Signal Peptide-Dependent Protein Transport in Bacillus subtilis, a genome-based survey of the secretome, unquote, where the researchers found that the Bacillus subtilis, a bacterium, has a natural capacity to secrete a diverse array of proteins into its cellular environment. These cellular secretions are very important when it comes to intracellular communication and can influence a wide range of biological processes, including tissue repair, inflammation, and cell growth. Specifically, the secretome includes a variety of signaling molecules such as hormones, growth factors, cytokines, and extracellular vesicles, which are released into the extracellular space to mediate interactions between cells. And you can think of this extracellular matrix as a fluid or gel-like substance that fills the spaces between cells in tissues. This fluid aspect of the extracellular matrix allows it to act as a medium through which nutrients, gases, and even waste products can diffuse between blood vessels and cells, facilitating essential biological processes. Or if you're using a petri dish, for example, it's just this space of fluid that contains the nutrients and a substrate for chemical reactions to occur. So in simpler terms, you can think of a secretome as a cell's way of sending out messages to other cells, telling them what to do, like healing damaged tissues or just growing new cells. Now, many of you have probably heard about exosomes. When comparing secretomes to exosomes, it's important to note that exosomes are just one part of the secretome. Exosomes are small vesicles that cells release to transport genetic material and proteins to other cells. They are essentially one of many ways cells communicate within the secretome framework. Saying secretome framework makes it sound like it's some sort of tech stack with a unique API to it. But anyway, so the exosome is a type of secretome that may be harvested and used for specific purposes. And right now, there are companies that are trying to innovate on this delivery mechanism, sending messages to hair follicles that are weak or dormant, and trying to get them to react to the exosomes that they're using. Maybe they're getting exosome from dermal papilla cells or hair follicle stem cells, and the purpose is maybe those exosomes have some sort of properties that can signal to the dormant hair follicles to, hey, start growing again or start growing better hair. Now, this is a very spotty area with minimal successes. Nevertheless, research is still being performed, but I know there are some clinics out there that are like, hey, we have special exosomes that, that can grow your hair. I mean, always be skeptical about these things and actually look at what those exosomes are, how they're harvesting them, and don't become a sucker. But in any case, it's still somewhat of a developing therapy. But by no means is it standardized. In the context of regenerative medicine, secretomes from stem cells are particularly intriguing because they harness the power of stem cells without the complexities and even some of those ethical concerns associated with using the stem cells themselves. Stem cells are unique because they can develop into different types of cells in the body, and their secretomes reflect this versatility. And maybe even you can find secretomes that are capable of producing those stem cells. So you're kind of playing with two different things here. You can have the secretomes of the stem cells that the stem cells themselves actually produce, right? Maybe that can have some sort of influencing factors on some tissues. Or you can have secretomes that signal out there into the environment, maybe through some sort of unknown process, and that generates new stem cells that further differentiates into specific tissues, right? All in all, either the secretomes of the stem cells or the secretomes that lead to creating stem cells, they are rich in regenerative molecules that can help repair and regenerate tissues, and that's very important for what we want to do with hair follicles and even hair cloning to some extent. So really, you could argue that finding the proper secretome or set of secretomes that lead to a specific kind of stem cell differentiation is the true goal here in the research. Or at least that's my perspective. And talking more about stem cells, there are various sources of stem cells, including adipose or fat tissue and mesenchymal stem cells. The adipose-derived stem cells are obtained from fat tissue and are prized for their abundance and ease of collection. They secrete a range of beneficial proteins that can promote wound healing and reduce inflammation. The mesenchymal stem cells, on the other hand, can be derived from bone marrow, skin, and other tissues. These sources of the stem cells, as well as their secretomes that influence said stem cells, are of important interest for their ability, again, to modulate the immune system and enhance tissue repair. And if you've been keeping up with the global hair loss development pipeline, you might be familiar with a particular company called Amplifica Holdings Group Incorporated, or for short, Amplifica, and their osteopontin-derived AMP303 synthetic glycoprotein molecule. For those of you who aren't familiar, osteopontin is a glycoprotein that plays a significant role in biomineralization and bone remodeling. However, the works of Dr. Maxim Plikus, a professor and researcher at University of California, Irvine, as well as a co-founder and chief science officer at Amplifica, discovered after examining hairy nevi, also known as skin moles, that the hair it produces contained senescent cells, and these senescent cells, after being mashed up by a blender and put into a mass spectrometry machine, certain proteins, or glycoproteins rather, were noted to be in abundance, which kind of gave a hint that, okay, osteopontin, as well as the molecule SCOOP3, seem to be involved in producing these long anagen hairs from these moles. Now, you can read more about this in the paper from Dr. Maxim Plikus et al., titled, quote, Signaling by Senescent Melanocytes Hyperactivates Hair Growth, unquote, published to Nature.com on June 21st, 2023. In a radical next step, Amplifica created a derivative of the osteopontin glycoprotein and named it AMP303, and in their early proof-of-concept clinical trial on human subjects, one single shot in the course of 6 months, and it was a single shot of AMP303, right, their proprietary derivative molecule of osteopontin, observed an increase in hair counts and also observed the transition of vellus hairs into non-vellus hairs. So that's really amazing. And the fact that it's only one shot, right? That's phenomenal. But obviously they need further, work and innovation to see what else is going on, right? Remember, in my interview with Dr. William Rassman, he is the chief medical officer of Amplifica, along with being Amplifica's co-founder. He mentioned that there were a lot of molecules that they recognized, that they found through Dr. Maxim Plikus' work. And just recently, if you go to the Amplifica website, they have expanded their pipeline and they're making it clear what they're looking for, right? We have AMP303, that's osteopontin derivative. We have AMP203 and AMP506. Now, I'm not sure if AMP506 and 203 are the same molecule, just with two different names, or they're separate molecules. But after looking at some online resources and past interviews that Dr. Maxim Plikis did with DermWire, AMP203 is stated to be an osteopontin derivative. So I think we can imply that AMP506 is also an osteopontin derivative. And finally, just from the words of Amplifica itself, AMP601 is a SCUB3 or SCUB3 synthetic glycoprotein derivative. So all of this is exciting. If you look at the pipeline itself, AMP303 is clearly further along in clinical trial testing. It got out of the lead selection, it got out of the preclinical development, and it already went into the first in-human, clinical testing. So that's pretty impressive, and hopefully we get to see more of AMP303. But who knows, maybe AMP601 being the SCUB3 derivative might have more promise. I don't know, maybe one of these things might be the winner here. But in any case, let's look at another study, involving secretome research. And actually, this study was pointed out by a Discord member of mine, a subscriber of mine, and they were like, hey, check this out. So we're going to be looking at this. It involves the use of secretomes and minoxidil. So this is going to be some interesting stuff. So I want to give a special thanks to Revan for actually pointing out this article that came out in the beginning of March. You notified everyone on the Discord server about it, so that's pretty cool. And I also want to thank David for actually helping getting the study as well, because it was paywalled and we had issue getting the study. And I also want to thank Zephyrius11, G Nathan, Avocado Gucci, and Alessandro for also helping as well, because it was a team effort. So thank you guys so much for actually getting the study and bringing the study to the attention of the Discord server as well as myself. It was greatly appreciated. So recently, a study came out by Lily Ligiawati and colleagues titled, quote, Hair Regeneration in Androgenetic Alopecia using Secretome of Adipose-Derived Stem Cells and Minoxidil, a comparative study of 3 groups, unquote, where a 3-arm study was initiated between a group that used a secretome of adipose-derived stem cells, a minoxidil-only group, and finally a dual minoxidil secretome group. The research employed a single-blind clinical trial method over a 5-month period, assessing the efficacy of adipose-derived stem cell secretomes, minoxidil, and their combined application. The evaluation metrics included hair density and growth with assessments conducted by clinical examinations, photography, trioscopy, and trichoscan. Now, the study revealed that each treatment method offered unique benefits. The adipose-derived stem cell secretome group, in particular, demonstrated remarkable effectiveness in enhancing hair density and promoting the initiation of the anagen phase, the active growth phase of hair follicles. Notably, this group achieved these results without inducing the common side effects associated with topical minoxidil use, such as the typical scalp irritation and dryness, which probably comes from the alcohol and the propylene glycol. But in any case, this highlights to us that the adipose-derived stem cells are a potent standalone option in the realm of regenerative therapy for hair loss, and it shows a great promise in both efficacy and safety. And furthermore, the combination of the minoxidil and the adipose-derived stem cell secretome group showed the most substantial benefits, so there's some sort of suggestion here that the treatments may have a synergistic effect. Table 3 provides an in-depth analysis of the specific outcomes of the study, and it reinforced the significant role of the adipose-derived stem cell secretomes. This treatment was particularly effective in increasing anagen and terminal hair growth, which are indeed crucial factors for healthy hair function. But there are some elephants in the room with this study, as it does have some noteworthy limitations and issues. The first being that this is a single-blind design. While, effective in some contexts, it can introduce some bias since only the participants were blinded to the treatment type and not the actual observers themselves. Next, we had a relatively small sample size of 60 male subjects, and of course that's going to limit the generalizability of the findings across different, demographics, particularly women who also suffer from androgenetic alopecia. And finally, maybe this might be a bit of a minor concern here because it is 5 months, but again, it is a pretty short study duration. Typically with, high quality or semi-high quality hair loss studies, you want to go for the 6-month mark, the 24, 25 weeks up to the 52-week or 1 year mark. That gives you a kind of a broader understanding of the full antigen cycle. But again, this is 5 months. Things could change if you added an additional month. Maybe you would see the, Minoxidil group possibly outcompeting the Adipose-Derived stem cell Secretome group. And finally, there might be some economic and practical feasibility issues concerning the large-scale production and application of adipose-derived stem cell secretomes. And, that wasn't really addressed. So if this were to be a promising treatment, of course it'll cost a lot of money, but I don't think that's really a problem for many people. If they really want their hair back that much, I'm sure they'll make the necessary financial commitments to try to, use something like this, if it indeed is a promising treatment. So if anything, this study kind of tells us that there is an impetus for further research in the matter of adipose-derived stem cell secretomes, and hopefully we get some more research on that that's properly controlled, maybe double-blind, randomized, and having 3 groups that we have here, maybe a 4th one being the placebo group of some kind. But all in all, that is pretty encouraging. Now, we're almost at the end of this video, but I just want to say, I want you guys to check out this paper. I'll leave it in the description or the comment section, and it's titled, quote, "The Paradigm of Stem Cell Secretome in Tissue Repair and Regeneration, Present and Future Perspectives," unquote, authored by Kate De Silva et al. And the authors talk about how secretomes are going to fundamentally change the approach to medical treatments, particularly in tissue repair and regeneration, by highlighting several innovative delivery mechanisms such as hydrogels, microspheres, and much more, right? The study shows us that these substances can be precisely applied to areas of damage. This targeted delivery is not only more effective, but also helps to limit the side effects often seen with treatments that impact the body systemically. And the implications for hair loss treatments are particularly promising. Imagine using a topical formula that carries some kind of secretome derivative, or maybe some secretome mimetic of some kind. I don't know, because secretomes can be pretty big, right? So I guess what I'm thinking of is like a glycoprotein of some kind, right? That can, modulate the CD44 receptor, like osteopontin or AMP303 is promised to do, right? So imagine we have something like that, where it's in some sort of topical formulation, and you can use it whenever you have either hair loss or some kind of weird scalp inflammation or whatever, right? Or maybe when it comes to hair cloning, we can find the proper set of secretomes or maybe a singular secretome that can help us produce viable cloned hair follicles. All of this is very, very promising. But I don't want to talk about this for too much. Again, go check out the description or the comment section for more information. I should be pinning a comment of some kind. And yeah, thanks guys for watching this video and being a subscriber to this channel. Anyway, see you on the next one. Peace out.